The next talk, presented by Gael Dur. Yeah? And entitled Mobidyc, Exploring Copepod Population Dynamics More Easily. Okay.
Yeah, perfect.
Okay, so
It's all good. No, I won't need it.
You don't need it?
Yeah,
Okay.
I'm good. Thank you.
Okay.
Hello. Let's go back.
Oh.
All right. Good afternoon, everyone. So my name is Gael Dur. I'm the head of the DGTalAquaLab from Shizuoka University. Yeah. And we have seen many talks today showing how innovative techniques can enhance our understanding of Copepod. And I'm going to follow that line by introducing individual-based model and a platform to develop such a model. But first of all, let me start with a personal story that underscores the importance of individual-based model. When I was a student, a PhD student, I helped one of my lab mates with her research. We went to the south of Taiwan to study Pseudodioptermus anandaeli. And her study was focusing on the effect of temperature and salinity on the development of that copepod. Now, she used an individual-based experimental approach, where you basically follow the growth and stage-specific progress of every individual at different conditions. And we were dealing with a tropical species. So definitively, our frequency of observation, 3 to 4 hours, every 3 to 4 hours. So if you have done this experiment, you have an idea on how intense this is. And so she tackled like several conditions, 3 temperatures and 3 salinities. And she recorded the entire development as well as the lifespan of the individuals, which lasted for over 2 months. So you can imagine how tedious and demanding this experiment was, right? And at the end of the experiment, we were more or less like that. So now let me ask you a question. Besides getting her PhD with those data, how many papers did she publish? 0. Yeah, what? Indeed. This is, yes. You don't, I mean, in terms of return on investment, you don't need an MBA to understand that this is not a good deal. And that won't push any other student to actually conduct such experiments, which is a shame because they provide invaluable data and information at the individual scale. And that should produce, at least as I heard, 1 paper, at least. Now, however, the downside of such approach is the scalability issue, right? You got good information at the individual scale, but extrapolating those results to larger population is a little bit tricky. And this is where individual-based model becomes invaluable. In contrast to mechanistic or differential model, which represents system-wide process and average behavior, the individual-based model simulates the action and interaction of autonomous organisms to study the dynamics. Now, individual-based model consider the uniqueness of the individual or the agent, their local environment or the local interaction, and third, their independent behavior. What I mean by being unique, you all study copepods, so you all know that they have different life stages and different predation pressure depending on their life stage. They have different size, they have different location, different resource reserve and history. acting locally means that all agents are not interacting with everybody in the simulated environment. They only interact with their neighbor. And being autonomous means that they do their own thing. They act independently and they have their own objectives, every individual, depending on their life stage. Now, all of these features are very important because they lead to the emergence of dynamics. So IBM allows you to study how a population dynamics emerge and is linked to the characteristics and action of individuals. When I was introduced to this approach, I was like, whoa, that's so cool. I love it. But like many students in biology, I was lacking proper training in coding, which basically prevents me from developing my own model, or I had to really study coding in order to do so. Fortunately, I was introduced to another platform called Mobidyc. And Mobidyc stands for Modeling Based on Individual for Studying the Dynamics of Communities. It's a multi-agent simulator for modeling population dynamics. And it was initiated in 1996 by a French researcher, Vincent Ginot, from the INRA, with three main goals: reducing or minimizing the demand for programming skills, supporting biologic-specific concepts, and be user-friendly to biologists. Now, Vincent worked a lot on this, and he had a lot of features, which leads to having Mobidyc operational in 2002. And this is how it looks like. So for example, here, typical example in modeling the predator-prey model, right? Where we have here I use wolf and goat, but you can use, of course, copepod and fish larvae, all right? And what you can ask, or what you can try to answer with this model is, how long would it take for those three wolves to actually hunt and eat all those goats or fish eating copepods? Each wolf and goat have some specific individual abilities, escape abilities for goats that are different between each other, and predatory ability or hunting abilities for each wolf. So you can run the simulation. It can run really fast, and you can slow it down a little bit. And the answer will be, well, you need to act really quickly. If you see some wolf in your area, you need to take your goat to safety before 4 months, otherwise you ended up with only 2 goats. There you go. Now, up until last year, Mobidyc was still used for scientific research, but mainly by my team. And I will present some case study of copepods, starting with Carol's favorite, Erythema affinis. All right. From the Seine estuary, what I did is using my lab mate, David De Vreeker's data on the temperature effect on the development and reproduction. And with Mobidyc, I developed 2 individual base models. One to reproduce the typical or atypical reproductive cycle of egg-carrying copepods, and another model which implemented this reproductive model into the entire life cycle. Then implementing also the effect of predation on top of temperature, I could simulate the seasonal cycle of Erythema affinis in the Seine estuary. I could also test the effect of global warming on this seasonal cycle. Let's move to another example in Korea with Paracyclopenis nana. This copepod is a coastal copepod and can sometimes be trapped in tidal pool. And my friend and colleague Jiwon did some experiments on UVB radiation on the reproductive cycle of that copepod. So I basically used her data, implemented it into the reproductive model, and run some simulations at a wide range of UVB radiation, so that I could estimate what would be the consequence of a post-exposure effect on UV of the production of many females. The third example is Apocyclops roe, species used in aquaculture in Taiwan. And in Taiwan, they cultivate it outside pond, outdoor pond. So they are very susceptible to change in environmental conditions. And where is Dr. Pan? Where's Pan? Where's Yan Lo? He's not here. Oh, he's over there. All right. So thanks to him, we conducted some experiments on the effect of salinity on the reproductive cycle of Apocyclops roe. My student, which is on the back over there, performed the parametrization and ran the simulation with many salinities, which allowed us to identify the optimal salinity for production for the species and what would be the consequence of a drop of salinity due to heavy rains, for instance. For the fourth and final example of Mobidyc application on copepods, I choose the invasive copepod species Arctodiaptomus dorsalis in the Philippines. And one of the hypotheses for its successful invasion is considered to be its high production ability when food is abundant. So using data collected by Dino Tordesillas, not here, all right, on the food effect and temperature effect on the reproductive cycle of the species, another student of mine could estimate the monthly production of a cohort of 1,000 individuals, female, under a variation of survival. And even at the highest survival rate and the highest food concentration, the production of this copepod is not particularly high compared to other species. Therefore, that suggests another factor beyond production rate contribute to the invasion of that species there. In total, we have 12 scientific papers published with Mobidyc. Not only on copepods, we also have some studies on fish and harlequin fly, tackling scientific questions in the field of ecology, biology, ecotoxicology, climate change, marine resource management, and aquaculture. Now, there is a downside. That's too beautiful. The creator actually passed away in 2007. And since then, the platform management stopped due to this dramatic event, leading to some problem of scalability and update. Scalability. The platform runs on the 32-byte VisualWorks platform, meaning even if you have a supercomputer with a lot of memory, you can only use part of it. Meaning that when you're trying to run a model that is complicated with many individuals, you run into that problem. And it only runs on old OS. Not so cool. So I worked with a Japanese computer scientist to develop the successor of Mobidyc. We call it Mobidyc. The idea is reconstruct the multi-agent simulation system with the same design rationale and use modern technique. We build on an open source called Smalltalk, called Faro. We use declarative language to make it easier to build the model. We also have some type and unit checking, and everything is done actually just to make sure that you can reproduce the model easily, review it, and validate it by other researchers. Running on 64-byte Faro, no problem of memory. Running on the most recent OS, no problem. Now, how it looks like. That's how it looks like. And for this type of model, when you're simulating an agent, it's defined by attributes, characteristics, and task. What is it doing? Okay. And the task, you actually pull them from a library, meaning you don't need to start from scratch, and if this reduces error, right? Now, I would like you to pay attention to the speed at the bottom. I modified it. The speed is now just in kilometer, just the distance. And the text changed to red over there. That's Mobidyc telling me, you're wrong, buddy. There's something wrong there. And if I change it to a proper distance by time, even if I change the unit, no problem. Mobidyc convert everything into international system of units. An example of the type of model that you can create with phytoplankton, copepod, and fish larvae. This is theoretical. And for a more practical approach, I invite you to go have a look at our poster P37 of my student. She developed a model of Eojaptamus japonicus in Lake Biwa to try to clarify why these copepods have seen this seasonal cycle disrupted for many years. So I hope I convince you that conducting individual-based experiments can not only lead to excellent knowledge at the individual scales, but also with the help of individual-based modeling and the proper tool, Mobidyc, which is free, by the way. You will be able to add more value to your effort and answer questions at the larger scale. Please go to GitHub, download Mobidyc, install it, test it, and if you have any question, I'm here. Thank you.
Thank you, Yael. Very inspiring talk. And we don't have time for questions, sorry.
Yeah, I'm sorry about that.
Yeah,
I'm free whenever you want. Okay. Thank you.